I think most important for a scientist, you have to listen and you have to read. And then you also have to try to understand the industry needs for the next generation. So that's the industry needs for the next, not the current, because the current is only just problem solving. So whenever I try to do a research project with the industry, I didn't want to actually address a problem of today. It has to be the problem of tomorrow. Because then is also another thing. If they have solved the problem of today, they are not interested anymore in you because they have to solve the problem.
What if chromatography could shift from diffusive to convective flow, and we could leverage the full potential by going all the way to the physical limit and revolutionizing how we think about mass transfer? Well, welcome back to part 2 with Alois Jungbauer, who is a professor emeritus of downstream processing at the Institute of Bioprocess Science and Technology at BOKU University in Vienna. He's also the scientific advisor of BioChromatographix International. We've covered the technical evolution. Now we are exploring the breakthrough technology that could change everything. It's monolithic chromatography, which is just not another media. It's a fundamental reimagining of separation science for larger modalities. Alois and his team are pioneering next-generation solutions that meet the complex demands of modern biotechnology, offering cutting-edge performance for gene therapy, vaccines, and advanced therapeutics. Let's dive back in. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. And now, speaking of a better way to do chromatography, let's focus on the work you're currently doing at BioChromatographix. Can you tell us what new resin or new technology you're developing at that company?
I question always myself, have we reached the physical limit in chromatography? I think if we come to very small molecules, then we are very close to the physical limit. But with the new gene therapy vectors, viruses, also exosomes, extracellular vesicles, all this material, which are in the 100 to 150, 200 nanometer range, we have not reached the physical limit. So the problem is with conventional chromatography, we would have a very low capacity. With the membrane chromatography, what I mentioned before, we have a higher capacity, a higher throughput, but no resolution. And material from BioChromatographix has features, we call it inverted morphology, flow morphology, that we can combine a high resolution and a high capacity. And therefore, we also work on what they call AXISFLOW, always on axial chromatography. And so far, the first results we have, the company is pretty young. We have to think about, they started last year in fall, that this material really has these promising features.
So how does then the AXISFLOW look like? Are these channels like very...
No, the AXISFLOW is also, they are channels. These are channels, and you could think about that is like inside out structure, that we have actually hollow spheres putting together, which are interconnected with spores. So in a conventional chromatography, you have material which is bead with pores. So that's, you have this pore structure. But once you have this bead with pores, it's not possible to have such large pores that the virus or gene therapy vector can actually diffuse into. Therefore, our strategy was, we have hollow spheres. These hollow spheres are also packed into the column, and the spheres are interconnected with pores.
And the virus or the viral vectors, they go into those larger pores then in your technology?
They go into these larger pores and have the possibility to bind out to the surface. We still have a much lower surface compared to a chromatography bead, but this is hypothetically, because in a chromatography bead, the real surface where the virus could bind is extremely low, because this would be only at the outside of the bead.
And what are the mechanisms involved in the binding?
The mechanism in the binding, so far our idea is, and also is, to have ion exchange, hydrophobic interaction, affinity chromatography. You even could do displacement chromatography. So the mechanism, the chemical functionalization is actually the same as with regular chromatography material.
And just out of curiosity, would this new technology also be interesting for traditional molecules, or would it be too complicated? Or no, like actually the current bead resin work much better for smaller molecules or antibodies or so?
I think we have targeted for large molecules and not for smaller molecules. That's definitely what we have.
Because there's no significant benefit for smaller ones.
There would be no real advantage. So I think there is not a chromatography material available which fits all purposes. Yeah.
And what other modalities do you think could be interesting for this new technology beyond cell and gene therapy? What have you in the plans?
I think beyond a cell gene therapy, there would be also the exosomes, extracellular vesicles, this type of things. Also, what is called protein superstructures, for instance, that's what we have in mind. Also, if there is a necessity, lipid nanoparticles, this type of, let's say, upcoming new modalities.
And to what extent would this be interesting to run in a continuous mode?
Then it would make a sense once you really start to produce gene therapy vectors in continuous mode. So far, I have not seen a lot of companies who are actually producing gene therapy vectors in continuous mode. Especially you have to think about when the virus is actually secreted or bodied from the cells and the cells get lysed, then the upstream processing get much more complex. So you would need a two-stage fermentation and so on and so forth. So we are not yet there in this field. There are first other problems to be solved.
And what are those major challenges you're currently tackling with this technology?
One of the major challenges is the expression titer in gene therapy vectors. They're still very low, especially for those applications where you need a very high dose. And so you have seen some of the even licensed products have been withdrawn from the market because they were not affordable. And here, manufacturing and manufacturing science plays a really important role to substantially reduce the manufacturing costs.
And what will be the next steps for your resin? What future development steps do you have planned?
I think the next step is what we have in mind, and this is actually what I really like, because this actually was in my head for many, many years, is also that to be able to scale it up. So you could make a really large-scale chromatography material with the same properties, same separation principles, and so on and so forth. So that would be then the next step. Because this is what then the companies would at least ask this, can you scale it up? Because otherwise they would not be interested to test that.
How easy would it be to scale it up? Is it quite a linear scale up, or would there be some?
There would be a linear scale up. So you can scale up in actual flow, really large scale. You have to understand that we don't want to disclose how we do the manufacturing of the material to get to this large scale, because this is our trade secret, but we'll be able to scale it up like a regular chromatography column, and the scale-up criteria is constant residence time, and that is known throughout the industry.
Excellent. That's exciting times definitely coming up. I would like to touch upon another topic because I remember your talk at the ESAC last year in Edinburgh when you talked about sustainability. And I think that's a very important topic when it comes to downstream processing. So tell us a bit what is on your heart and why is that such an important topic and what can we do about it as an industry?
I think the sustainability as such is also important, especially the water consumption, because to have enough high quality water would limit the sites at our globe where you can manufacture even biopharmaceuticals. Because in a lot of regions on our globe, we have a water shortage. So therefore, that's, I think, the mere scale of, let's say, savings compared to other industry sectors like food industry and so on is pretty low. But once we think about distributed manufacturing, if we are not able to actually reduce our water consumption, we can forget about distributed manufacturing. That's, I think, the main point. Of course, find water saving, but a little bit of water saving in our households would outpace the water savings of a pharmaceutical industry by far. So that's it. But I think this sustainability issue also is important because it goes along with the reduction of cost of goods. And of course, if all industries start to save water, at the end, we will have also a global impact. That's clear.
So to understand correctly, Alois, so if we reduce the water consumption, we could significantly save in cost of goods. Is that what you're saying?
Yes. This substantially reduces the cost of goods.
Is this because of the water treatment that's involved?
Water has a high impact to produce the water. There's a lot of impact to actually keep the water in this high quality in a pharmaceutical industry. And then the second is, if we reduce the water consumption in a bioprocess, the process itself gets more compact. And when the process gets more compact, we reduce the floor space. And then when we reduce the floor space, a lot of energy and resources are saved for, let's say, air conditioning and providing high quality air.
Before we continue, Biotech founders, I want to talk directly to you. Do you know that feeling when an investor asks about your manufacturing strategy and you freeze? One wrong CMC decision can cost you $2 million and 18 plus months of delays. Yet many CEOs are flying blind on these critical decisions. That's why I created the 1-week CMC strategy assessment. In 1 week, you get a personalized assessment of your molecule and development stage. We identify critical gaps before they become million-dollar mistakes, plus a strategic roadmap for high-stakes investor meetings. One CEO told me, David took me from feeling powerless about our CMC roadmap to confidently pitching investors. An absolute game changer for time-pressed CEOs. Whether you are preparing for Series A, approaching IND filing, or facing scale-up challenges, This gives you confidence to ace investor and board conversations instead of surviving them. Ready to stop second-guessing your CMC strategy? Check the link in the show notes and secure your spot. Because your breakthrough therapy deserves a solid CMC strategy. Now, let's get back to our episode. That's very interesting. Wow, very interesting. In this last part of our conversation, Alois, I would like to circle back to the beginning when you were talking about your career. And as you're now looking back, or I would also be curious to understand, because you have been able to consistently either surf on trends or even anticipate trends in the bioprocessing industry. And now again, you're working on a new technology, on the monolithic technology. What helps you to see these opportunities, perhaps even before some others see that in the industry?
I think most important for a scientist, you have to listen and you have to read. And then you also have to try to understand the industry needs for the next generation. So that's the industry needs for the next, not the current, because the current is only just problem solving. So whenever I try to do a research project with the industry, I didn't want actually address a problem of today. It has to be the problem of tomorrow. Because then is also another thing. If they have solved the problem of today, they are not interested anymore in you because they have to solve the problem. Then to think about what is the next generation of pharmaceuticals or biopharmaceuticals. And so far, it seems that there is a huge, let's say, a potential in this gene therapy or around the gene therapy, gene editing, and so on and so forth. So that's really a huge potential. The question is, how fast it will go, and is actually can the companies really make a profit out of that? Because you see, why was this hype of gene therapy in the 2018s and 90s? A lot of investors want to invest in tech utopia. And 2018, gene therapy was tech utopia. Everybody think, okay, we will do, everything will be cured by gene therapy and so on and so forth. Then the misunderstanding that we can actually produce this material, let's say, inexpensive way, which is not true. Because for a viral vaccine, you only need a fraction of the dose of viruses compared to gene therapy. And that was the big misunderstanding. So currently we are a little bit in a depression, but I'm sure we'll come up with the comeback once we have clinical successes again.
Now, besides listening and reading, what other advice would you give a young scientist who is starting their career? Because let's face it, we live in a fast-changing world. We have new technologies, we have AI, we have machine learning, we have new development, cell and gene therapy, and it's never ending. So what would you say, and what have you told your PhD students actually? What are good habits to develop?
So the good habits to develop, I think the most important point is that you love this discipline. And then, of course, with that, it comes a certain, let's say, intuition. So these things cannot be planned. This is for a young career. Sometimes it is you have to be at the right time, the right place. So that's important. What I would recommend also, if you go as a young scientist to institution, whatever industry, academic, and you don't have a mentor, then leave it. Because this is also very important.
Yes, having a mentor is extremely key.
But if you actually address the right problem, sometimes this is mere luck also. But also to have a certain persistency. A persistency not to hop on, hop off if there is, like, say, something new. So really try to work, even there is a certain depression in a field, continue to do this.
Before we wrap up, Alois, what burning question haven't I asked that you're eager to share with our biotech community?
I think a lot of people ask about nowadays the sustainability. I think one question would be definitely in the biopharmaceutical industry, the affordability. When there is now more and more a gap between actually development of pharmaceuticals and what our healthcare system can afford. So that is a societal problem. And the other thing, and this is really more of a political problem, is that in our society we have now also an adverse, let's say, ideas against science. That is something worries me most. Thinking about yesterday, day before yesterday, it has been shown that they have now polio cases in Germany.
Oh, really?
Yeah. There is no need to have this. So we thought about we could wipe out this disease. So this is something which goes beyond, let's say, science and engineering, but impacts it a lot.
And beyond science, if you look now back on your career path, what is the legacy you want to build right now?
The most important thing is in our society is that science and engineering helped us that we are at the stage what we are currently. And my impression is scientists and engineers are not really appreciated by the society. So look to the news wherever you be, you have celebrities and so on and so forth, and then ask yourself, what have they contributed to the benefit of the society, except wearing certain dress or whatever. So the impact of our engineering and science is that we have a good quality of life, healthcare, communication, and so on and so forth. There's only one newspaper in the German-speaking region which really appreciates science very well, and this is the Neue Zürcher Zeitung.
That's a good newspaper, definitely.
And I like to read the science section of this newspaper, because when they write something which is in your field, then you can actually get an idea about the quality of the writing. And every time they write, I think, this is really cool. I think, oh, these are really excellent science publishers.
Excellent. Wow. Thank you for sharing this thought. This has been such a great time, Alois. Thank you for sharing your insights, your passion. What is the number one takeaway you want us to walk away with from this conversation?
The number one takeaway is that actually in the field of downstream processing, and especially with gene therapy vectors, we have not reached the physical limits yet. And there is room for improvement.
Excellent. There's room for improvement. And that's why we need each scientist, each one of you who is listening, because science is important. We have heard it now. So thank you very much, Alois, for sharing all this, your wisdom, and for being on the show today. So where can people get a hold of you?
I go usually to these conferences, Bioprocessing Summit, Bioprocessing International. If they accept me, I like to go to Recovery Conference. I'm co-organizing or co-chairing the next Integrated Continuous Biomanufacturing Conference in October in Dubrovnik. I have been invited for a talk at Advancing Cell and Gene Therapy next year in North Carolina. So if they go for listening, they can get hold of me.
Fantastic. So smart biotech scientists, go to these conferences, talk to Alois, and let us know what you think about downstream processing and what you think is the next thing. So thank you again, Alois, for being on the show today. It was a huge pleasure.
No, it was a pleasure also for me.
What a fascinating journey through the past, present, and future of downstream processing with Alois Jungbauer. His insights on sustainability, continuous processing, and emerging technologies are game changers. Remember, if you are wrestling with process development or any manufacturing questions you may have, book a free call with me. I am here to help. You find all the links in the description. And don't forget to leave a review on Apple Podcasts or your platform of choice. It will mean the world to me. Thank you so much for tuning in, and I'll see you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.